Hello everyone, my name is Somkiat Putinath, and in this presentation I'll be talking about my project, Single-Cell Transcriptomic Analysis of Microglia in Alzheimer's Disease. Alzheimer's disease, or AD, is a progressive neurodegenerative disorder characterized by impairments in memory, language, as well as visual, spatial, and executive functions. It is the most common type of neurodegenerative disease and the most common type of dementia, affecting over 40 million people worldwide. The main pathological hallmarks in all AD are accumulation of senile plaques, which are extracellular aggregates composed largely of amyloid beta peptides, and formation of neurofibrillary tangles, which are intracellular inclusions rich in hyperphosphorylated tau protein. While there are pharmacologic treatments available, these options only ameliorate symptoms and the disease inevitably progresses. There is also a lack of quality evidence supporting any effective preventive strategies, and even after decades of studies, the underlying pathomechanism remains unclear. Perhaps a better understanding of the disease's pathogenesis will aid in the discovery of better therapeutic measures. There are two main types of AD. The first type is called familial or early onset AD, which represents about 5% of all patients and is mostly dominantly inherited. This form of AD occurs in people aged less than 65 years old and is associated with some of the causative genes listed here, with PSEN1, PSEN2, and APP being the most studied. Amyloid beta production appears to be the primary cause of this form of AD. Another type of AD is called sporadic or late onset AD, which represents about 95% of all patients and occurs in people aged over 65 years old. Genome-wide association studies, or GWAS, have identified numerous genes associated with this form of AD, but exactly how these genes drive AD progression remains largely unresolved. Defective clearance and aberrant amyloid beta degradation are likely important drivers of cases with this form of AD. Many previous studies focus on the amyloid cascade hypothesis, which proposes that AD is initiated by amyloid beta deposition, which leads to tau pathology, synaptic dysfunction, inflammation, neuronal loss, and ultimately dementia. The amyloid cascade hypothesis is also linear in the sense that it considers amyloid plaques as the direct cause of progressive neurodegeneration, where amyloid beta is the major constituent. Amyloid beta has been found to exert its effects through many different mechanisms across different cell types, and this highlights the importance of expanding the view towards this disease beyond a neuron-centric one. There are indeed alterations in other non-neuronal brain cells which have been observed and are suspected to be associated with AD pathogenesis, and they will be discussed subsequently. In addition, this hypothesis proposes a quantitative model in which amyloid beta buildup is causing the disease, implying that lowering amyloid beta plaques or oligomers is sufficient to halt the disease progression. This model fails to explain some of the current findings. For instance, in sporadic AD, there is a discrepancy in amyloid load and disease symptoms. In familial AD, PSEN1 mutation, which is one of the causal variants of the disease, doesn't actually increase amyloid beta, but actually destabilizes its three-dimensional structure and causes pathological effects. So there appears to be a qualitative rather than a quantitative effect of amyloid beta proteopathic stress on the pathogenesis of AD. An alternative theoretical construct called the cellular phase of AD has been proposed to better explain the silent incubation period of the disease. Here, the preclinical or prodromal phase of AD is attributed to the compensatory mechanisms to maintain normal cognitive function. The disease commences insidiously once pathological reactions are initiated in various cells of the brain, including non-neuronal cells. Responses observed in these cells may actually be benign or protective, but ultimately becomes devastating and lead to irreversible dyshomeostasis of the brain. The biochemical phase of AD is characterized by formation of various amyloid beta and tau conformations. Initially, there's abnormal amyloid beta aggregation and amyloid precursor protein processing. In addition, there's also tau tau conformer formation, which is followed by hyperphosphorylation. There is subsequent seeding and paranoid propagation of these abnormally folded proteins, as well as generation of oligomeric amyloids and generation of amyloid plaques and neuronal tangles. The different amyloid beta and tau conformations bind to normal amyloid precursor protein and tau protein, and they disturb the normal activities of these proteins in neurotransmission. And they also exert proteopathic or aggregate stress on the brain cells by interacting with the proteins and membranes, as well as interfering with the signaling and other functions. The key regulator during the proteopathic stage is the lysosomal and endolysosomal system, especially autophagy. But in spite of all this, the effects of proteopathy shouldn't be considered overwhelming or irreversible in the beginning. This is because the brain cells are able to survive under such stress for many years, and the initial responses observed are physiological and is aimed towards maintaining homeostasis. The critical phase occurs when there is a transition from early and reversible response to a chronic, irreversible, and cell-autonomous one, which is no longer dependent on the initial amyloid beta load and the tau aggregate stresses. In sporadic AD, amyloid beta and tau accumulations may be a sign of increased proteopathic stress and should be considered as risk factors for disease progression. However, additional cellular factors are determinative in the evolution towards dementia. The real cause of sporadic AD may be upstream of the proteopathies and is likely manifold. So, aging is likely the major driver of pathogenesis in this group, as it is the greatest risk factor for sporadic AD. Nevertheless, proteopathic stress during the biochemical phase results in cellular phase of AD, which we will now discuss. To better illustrate the concepts underlying the cellular phase of AD, we should look at some of the pathological changes observed within each cell of the brain. First, the primary component of the brain vascular system is the blood-brain barrier, which primarily consists of the vascular endothelial cells, pericytes, and escherichia and feet. Vascular defects are suspected to precipitate AD by causing hypoperfusion or hypoxic stress. In addition, blood-brain barrier breakdown results in many detrimental consequences, including accumulation of neurotoxic serum proteins in the brain, inflammation, vascular and synaptic dysfunction, defects in amyloid beta and tau clearance and metabolism, as well as vascular problems such as cerebral amyloid angiopathy. Second, neuronal action to tau and amyloid beta proteopathic stress is complex and not merely just simple cell death. For example, brain dysfunction is mediated by decrements in synaptic plasticity, which disrupts neuronal connectivity. Abnormal brain circuitry is also induced by amyloid beta stress, which is manifested by evidence of excitatory non-convulsive seizure activity in the neuronal networks. Third, astrocytes are by default intimately involved in amyloid beta catabolism, as well as synaptic processes such as modulation of synaptic output, synaptic pruning, and synaptic formation. In AD, reactive astrogliosis is observed where astrocytes undergo complex and dynamic response to brain damage, which are characterized by astrocyte hypertrophy and proliferation, as well as augmented expression of various intermediate filaments. Whether this response is beneficial or harmful remains an open question and may depend on the stage of cellular phase of AD as well as the affected brain region. Fourth, microglial alterations lead to inflammatory responses, which include complement activation, increase of inflammatory cytokines in the cerebrospinal fluid, as well as increase in the reactive oxygen species. Genetic data unequivocally place inflammatory responses as an important finding in AD, as GWAS studies have reviewed many susceptibilities loci which have been ascribed to the innate immune system regulation. However, a similar problem is encountered where it remains uncertain whether these responses are beneficial or damaging to the brain. Finally, oligodendrocytes, which represent the largest group of non-neuronal cells in the brain, may be associated with neuronal vulnerability to AD through the process of demyelination. This is the conceptual framework which summarizes some of the key concepts mentioned earlier. It is proposed that AD pathogenesis begins with the biochemical phase, which is characterized by alterations associated with abnormally folded amyloid beta and tau protein, and subsequent generation of amyloid beta plaques and neurofibrillary tangles. While the effects of proteopathic and aggregate stress exerted by these entities may be mild and reversible initially, they can elicit compensatory mechanisms during the cellular phase, which can then turn into chronic, irreversible, and pathological processes. At this point, chronic inflammation and cellular fatigue is observed, and the disease progresses inexorably towards the clinical phase, with marked neurocognitive decline. The cellular phase highlights the stages of disease transition from an early and benign physiological response to a detrimental and pathologic one. Interactions between different cells of the brain during this phase contributes to AD progression. At this point, it appears that there is a lot of gap in knowledge regarding the underlying contribution of each cell type in the brain and their interactions with one another towards the pathogenesis of AD. Transcriptomics is a useful approach to studying this question, as it captures the genetic expression profile of a particular specimen, from which conclusions regarding its phenotypic characteristics can be drawn. A powerful technique in transcriptomics is RNA sequencing, where transcripts in the RNA extract are captured and quantified through high-throughput sequencing. This allows analysis of the entire specimen's transcriptome. Previous studies are able to analyze cell type-specific gene expression profiles by isolating cell types using cell surface markers. One of the limitations in this method is the prerequisite of having known sets of markers specific to each cell type, and different cell types sharing similar sets of markers may also obscure analysis. Another limitation is the inability to identify subpopulations of a given cell group, since all of the cells are being sequenced in bulk. Resolving cellular heterogeneity is important, since cells within a given population may exhibit significant differences in gene expression profile and hence phenotypes. Single-cell RNA sequencing is a novel transcriptomic analysis technique where RNA sequencing is performed on individual cells. This method solves the limitations mentioned earlier, and studies using this technique have indeed identified subpopulations in each cell type of the brain and their associated biological pathways in AD. Further analysis of these cell subpopulations, as well as their roles in each stage of the cellular phase of AD, will be useful for elucidating the disease's pathogenesis. Since there is substantial evidence implicating biological pathways associated with microglia in AD, it may be useful to further explore how they contribute to the disease's progression. The objectives of this study are to explore the roles of inflammation and endocytic pathways in microglia in AD patients, explore the interactions between microglia and other cell types through coordinated gene expression on endocytic pathways in AD, and to identify microglial subpopulations from gene expression and link them to establish genetic risk factors for AD. All of the data used in this study will be obtained from an online database, and this is the list of studies from which our data will be obtained. So basically we include all of the studies that has single cell analysis for Alzheimer's disease that we can find. And data from studies in row 2 to row 4 will be used to correlate gene expression profiles across different cell types with clinical pathological phenotypes such as the patient's age, degree of cognitive impairment, Braak staging, etc. And the microglial subpopulations will be identified from the combined data of all of these studies, all of the studies listed here. For single-cell transcriptomic analysis, the first step is to pre-process each dataset by subjecting them to a series of quality control measures. Then iterative clustering strategy will then be performed to identify subpopulations of each established cell type. And in this process, only informative genes or highly variable genes will be included in the calculation of cell-to-cell similarity in order to minimize the interference from genes whose expressions are unchanged across brain cells. Correlation between gene expression, cell subpopulation structures, and the patient's clinical pathological phenotypes will be calculated to identify potential biomarkers. Biomarkers related to inflammation, esocliasis, and endocytic pathways, as well as other pathways identified through the differential expression analysis and the functional enrichment analysis, will be further investigated in depth in order to gain mechanistic insights of how they are involved in AD. Gene co-expression network and regulatory network will also be reconstructed to identify cooperation between microglia, astrocytes, and other cell types. Subpopulation specific markers will be linked to establish genetic risk factors for AD in order to understand the transcriptomic impact of these factors. So this is the end of the presentation. I'll leave you an open-ended question, which is, what is some of the advantages of single-cell analysis over conventional bulk tissue analysis? Thank you for listening to my presentation.